Hi, my name is second class Jill Corzini of the Massachusetts Maritime Academy, and in this presentation, I'll be teaching you about the tropical sea sponge Tectitethya crypta. Tectitethya crypta, formerly known as Cryptotethya crypta, is a species of demo sponge. It is one of the most diverse classes in the phylum of Porifera. They include about 76.2% of all sponges in the entire world. Their skeletons are made up of spicules that consist of fibers made up of both protein and silica, or just one of the two. They also consist of small, flattened tubercles that are about 2 to 3 millimeters in size. They are a dark black-green color. However, on the sea floor, they appear as if they are a whitish-gray color due to the sediment coverage on the seafloor. They also have a strong, firm structure that let them withstand storms and currents. What are sponges? Sponges are living animals. People often believe that sponges are plants rather than animals, and it's a common misconception due to some of the characteristics of the Porifera phylum. There are about 5,000 to 10,000 species. Most of them live in salt water. Only about 100 to 200 species live in freshwater. Sponges are the most simple multicellular animals in the world. There are two types of sponges, encrusting and freestanding. The Tectitethya crypta are encrusting. This means that they tend to cover surfaces and rocks. This is like moss. The diagram on the right shows the basic anatomy of a simple sea sponge. Their feeding habits. Sponges do not have mouths. Instead, they have small pores that they let water flow through to get their food. They are, in fact, filter feeders. The name for these small pores located along the sponge are osteas. This is how the sponge allows water to enter its body. There are then cells located in the cell wall that filter out food and allow the rest of the water to pass through the sponge. The water is only pumped in one direction through the osculum. This is known as the sponge's little mouth. Flagella along the body chamber allow the water to only move in one direction through the sponge and help push it through. The diagram on the left gives a simple representation of how this process works. This short video here shows how a sponge gets its necessary oxygen and food through water. It allows water to enter through the ostiums. They are then moved into the filter chamber. Here, the flagella push the water through the sponge and allow it to exit the body after the nutrients and oxygen are collected. Their location. As you can see in the diagram on the left, sponges are located around the entire world. They live in lagoonal environments and they live on sediment-covered seafloors. They are most commonly found in South Florida and throughout the Caribbean Sea, such as the Bahamas, Dominican Republic, Jamaica, and Belize. Reproduction. Sponges are able to reproduce both sexually and asexually. They asexually reproduce when pieces or fragments of the sponge's body are broken off and water currents then deposit them in a new location on the seafloor. This happens through budding and fragmentation, as shown on the right. Sexual reproduction occurs when males acting as sponge cells release their gametes into the water. Then the sperm cells travel closer to the female and the collar sponge cells of the female then allow the sperm to enter its body. Sponges are also hermaphroditic. which means that it is able to choose its sex based on its reproductive needs. Here is a simple diagram of how a sponge reproduces. On the left, you can see budding occurring as asexual reproduction happens. The bud is taken off and then deposited on the seafloor, and a new sponge is then created. On the right, you are able to see how sexual reproduction occurs. As the sperm cell meets the egg cell, it then is engulfed by the collar cell. This is where fertilization happens. The embryo then uses its cilia to propel itself through the water. It eventually will settle down and grow to be an adult sponge. So why humans care about sea sponges? The Tectitethya crypta is actually used for antiviral and anti-cancer drugs. It has been used to help treat and cure diseases such as AIDS, HIV, leukemia, herpes, pancreatic cancer, lymphoma, breast cancer, non-small cell lung cancer, and bladder cancer. Cytarabine is a synthesis inspired by natural products. Arabinonucleosides can be isolated by the marine sponge. The two nucleotides taken from the sponge are spongothymidine and spungoridine. These two nucleotides are used in drugs for DNA synthesis as well as chemotherapy. In this diagram, at the top, it is shown how the two nucleotides make up the cancer-treating drug cytarabine. This drug is used for chemotherapy patients with both lymphoma and leukemia. The diagram at the bottom shows the inhibition of DNA duplication as well as DNA transcription. What this means is that the cytarabine is able to go into the body and convert to cytarabine 5 triphosphate, which then creates a fake nucleotide incorporation in the DNA. This allows the cancer to stop growing. The elongation of the DNA strain then interrupts the process of the DNA synthesis. The diagram shown here explains the original natural products from the sea sponges and their therapeutic area in humans. The two that we are focusing on is the spungoridine and the spungothymidine. These are the two nucleotides that can be found in Tectitethya crypta. The spungoridine is used as an antiviral treatment. This is used often for HIV patients. The spungothymidine is often used for cancer treatment, most commonly in leukemia and lymphoma. In 1969, the first marine drug approved for cancer treatment was these two chemicals created from the Tectitethya crypta. Some other major breakthroughs from these chemicals is the HIV drug AZT and a breakthrough in AIDS treatment in the late 1980s. In years to come, biotechnicians hope to come up with new drugs to use against different cancers and other diseases. This has been my presentation on Tectitethya crypta. I hope you enjoyed and learned a lot. Thank you.